Okay, hi, my name is Jessica Martin, and today we're going to talk a little bit about sensory stimuli, and in specific, talk about chemoreceptors and the processes of olfaction and gustation, or smell and taste. So first up, what are chemoreceptors? Chemoreceptors are activated by the binding of ligands. And like I mentioned before, these type of receptors are responsible for detection of taste and smell. They are the oldest this is the oldest evolved way to get sensory stimuli. So this has been very evolutionarily protected. We have unicellular organisms that use chemoreception, and we also have evidence of organisms that had really rudimentary or no central nervous systems who also used chemoreception. So this has been around for a while. The first thing that we'll go into is olfaction, or our sense of smell. This picture here kind of shows a gross anatomy viewpoint of the nasal cavity and the system itself. The first step of smell would be the odorant molecules in the air entering the nasal cavity and dissolving on the inside of the nasal cavity, which is called the olfactory epithelium. It's very important that odorant molecules are dissolving. If they are not dissolved, they will not be able to be detected by the sensory neurons of the olfactory system. So we can go ahead and take a little bit of a closer look here. This is a zoomed in picture of the pathway and how our neurons are moving through it. So here, this bottom layer here labeled number 4 is the representative of the olfactory epithelium. And these here are our primary motor neuron, or primary sensory neurons. So the odorant molecules would bind to the dendrites here once they're dissolved, and the binding of the ligand would activate G olfactory, which is a G protein that would then activate on its pathway to open ion channels and depolarize the primary sensory neuron of the olfactory system. So once we have that depolarized, then the axon potential, or the action potential, will move down the axon here. And the axon of the primary sensory neuron actually is moving through bone here. This thing labeled number 3 is bone. And as it moves through the bone, it goes to the olfactory bulb. This is actually the cribriform plate of the ethmoid that it's moving through. But once it gets through past that, then we reach this giant yellow structure here, which is the olfactory bulb. And notice that we have synapsing here, and the synapses go from multiple primary neurons onto one sensory neuron. This is called convergence, meaning that we have a lot of stimuli that are then condensed into fewer neurons that are taking signals back to the central nervous system. So once we have those synapses onto the secondary sensory neuron, then they move down the axon to the olfactory cortex, straight to the olfactory cortex. Every other sense and their pathway to the cortex goes through the thalamus. However, for olfaction, that is not the case. It again just goes to show that this is a much older, more primitive yet effective way of getting sensory stimulus. Some cool things about the olfactory pathway is that some of the stimulus is also processed by the limbic system, which is why sometimes we smell things and have really strong emotional memories tied to them. It's because it also gets processed by the limbic system a little bit, and current research indicates that different primary neurons are responsible for detecting different ligands, and then that, though, depending on which neurons are fired and what information gets sent back to the central nervous system, the brain will cobble together what smell it's detecting. Okay, now let's go ahead and move on to gustation, or taste. Taste is a little bit different. There are more specifics that go along with it and more intricate pathways, just because there are different options based on the 5 taste sensations, which are here. We have bitter, umami, sweet, sour, and salty. Each of these are triggered by different molecules. So bitter, umami, and sweet are triggered by organic molecules, salty by sodium ions, and sour by hydrogen ions. Taste has a lot of evolutionary benefit and things that it tells us about our environment. Umami and sweet foods have been associated with nutritious foods in the past. Bitter foods have been associated with things that might be toxic that we might want to spit out. And we also have things like specific hunger, where if our body, for example, is low on sodium, then we might develop a craving for sodium to kind of fill that gap and return to homeostasis. So let's go ahead and take a closer look. This here is a really zoomed in picture of the tongue. And this onion-shaped structure is a taste bud filled with a bunch of different taste cells, which are the yellow cells here. And the taste pore is the opening at the top that would allow for a ligand to bind to a taste cell. There are 4 types of taste receptor cells. The first type is a glial cell, or supporting cells of the nervous system. The second taste receptor cell type is responsible for the transduction of sweet, umami, and bitter taste sensations. The third is for sour taste sensations, and the fourth is a type of cell that will help to regenerate neurons and taste receptor cells as needed. So it's sort of stem cell. So what would happen is we would get a ligand that would come and dissolve onto the tongue. Again, it's important for chemoreception to have the ligand dissolved before it's detected. And it would bind to the microvilli, which would then start a pathway inside a taste cell. For our taste sensations that are generated by organic molecules, they would bind to the microvilli and activate a G protein called gusteucin, which would then go along its pathway to allow calcium into the cell, causing depolarization. The calcium's depolarization would generate ATP, which would then be released onto the gustatory primary neuron. For what we know about sour taste sensation, we know that the hydrogen ion would enter through an ion channel and go through pathways directly to get calcium channels open and again depolarize. However, for the sour taste, we are using neurotransmitters to activate the primary sensory neuron. For salty, the verdict's kind of still out. We're not super sure the specific pathways that the salty taste sensation is taking. So we do have multiple options just because there are more things going on here. But once they are in the primary sensory neuron, then they are along the gustatory nerve to the medulla, to the thalamus, to the gustatory complex for decoding by the brain. In terms of chemoreception for the gustatory pathway, chemoreception does also occur in the stomach for gustation, which is important to note. It does use the same pathways, and it doesn't lead to taste in the traditional sense, but it does help to prep the small intestine for what enzymes it might need to release to properly break down the food that we have ingested. So it does still definitely serve a function in the stomach as well. Last thing here, I put together a little concept map. Let me move it so you can see the full thing. So right here, the broad topic we have is chemoreceptors, which branches off, of course, into olfaction and gustation. I have some notes here that are applicable to all chemoreceptors, and then, of course, some specifics on olfaction itself and its pathway, and gustation as well, and the different types of ligands they use and what that might mean. So that is my little presentation on chemoreception. Thank you.